That was a wonderful panel discussions that Ed led. So thank you everyone for participating in that sessions. I would like to now introduce our next moderator, Dr. Amelie Ramirez, who is the Director of Institute for Health Promotion and Research, as well as Chair of the Department of Population Health Sciences at UT Health San Antonio. Dr. Ramirez has been really wonderful in not only speaking for the sessions, but also will be moderating the panel discussion at the end. So welcome, Amelie.
Thank you very much, Tram. I really appreciate what NCI has done in really pulling this important session together. So we really appreciate it. This session is a kind of the second part of what we had from this morning or earlier today in terms of understanding Hispanic populations in health. We're going to have 5 presenters. And we have a short amount of time, so we're going to ask each of our presenters to please try to stay on time as much as possible. So I'm going to introduce you all at once, and then at the end of your presentation, I'll ask the next person to start. So in terms of our full panel this afternoon, we have Dr. Luis Carvajal-Carmona, who's professor of biochemistry at the University of California, Davis, and he will be talking about genetics and cancers in the U.S. Hispanic populations. Followed will be Dr. Douglas Morgan, who is with the University of Alabama, and he will be discussing the interplay of infection, co-evolution, and genetics in gastric cancer risk. And then I will be speaking about liver cancer in the U.S. Hispanic population, specifically looking at incidence and mortality trends. And then we will have Dr. Adam DeSmith, assistant professor at the University of Southern California, who will be talking about childhood cancer health disparities in Hispanic populations. And wrapping us up will be Dr. Frank Benedo, the Associate Director of Cancer Survivorship at the University of Miami and the Sylvester Cancer Center, and we'll be discussing cancer survivorship in Latinos. So again, thank you very much for pulling this wonderful panel together. And our first speaker will be Dr. Luis Carvajal-Carmona.
Good morning, everyone. I am Luis Carvajal-Carmona. I will be speaking today about cancer genetics in U.S. Latinos. I would like to thank the organizer for inviting me to present today. This morning, I will be covering 2 aspects. I am going to give you an overview on the genetic ancestry of Latinos, and then I will be covering 3 areas on cancer genetics, starting with monogenic disorders in Latinos. Then I will talk about GWAS and polygenic risk scores. And finally, I'll be talking about tumor genomics. This map showing the U.S. counties where most Latinos live. Most of them live in the Southwest, Florida, and the East Coast. On the right, I'm showing the main Latino populations in the country. Mexican Americans account for around 65% of all U.S. Latinos, over about 36.6 million of them. The second largest population is Puerto Ricans, who represent around 5.6 million people. We have a lot of Central Americans, and from South America, Colombians are the largest population with around 1.2 million. I am Colombian, and there are more Colombians in the US than in people in 9 states. So this indicates that Latinos are very diverse, and there are a lot of large subpopulations in the community. And here I'm showing data on the impact of country of ancestry or country of origin in the genetic ancestry of your Latinos. If Latinos have a Caribbean origin, as shown here on the right, the purple or African ancestry tend to be higher than if Latinos are of Central or South American origin, or Mexican, where the ancestry coming from American Indians and Amerindians is higher. Here on the right, I'm showing a table which has some data on birthplace status. If Latinos are born in the US, then they tend to have a higher European ancestry, and if Latinos are born abroad. This probably indicates some more recent admixture with the Europeans here in the country. I will now start speaking about actual cancer genetics, and I will start with the monogenic disorder. The most important characteristic of monogenic syndromes for cancer in Latin America and Latinos is the high prevalence of founder mutations. These are two examples of founder mutation studies in the region. In this table is showing results from a study led by Pilar Carvajal, where we have a contribution where we showed that a really small number of mutations accounts for 80% of the carriers of BRCA1 and BRCA2 in the country, in the entire country. Here on the right, I'm showing a study where we also have a large collaborations of multiple Latin American countries, Spain, Portugal, and an European colony in Africa, Angola, where we show that a single mutation, the most common throughout Latin America, has a single origin in Spain. This is very important because we can leverage these founder mutations to carry out penetrance studies and to consider new ways to prevent cancer via genetic testing and counseling in the region. Latino families are also very useful to identify new cancer genes. This is an example of a study that we published a few years back, where we discovered a new form of gastric cancer, a new familial form of gastric cancer. In part because gastric cancer is very common in the region, and also because the families tend to be large, so they facilitate monogenic analysis. And on the right, I'm showing after a few years, we published a review on this new syndrome. And actually, we believe that a couple of these genes are now emerging as bona fide familial gastric cancer genes, including PALB2 and ATM. So the discoveries did actually happen because we were studying these families from Latin America. I will now have a couple of slides on GWAS and PRSs in Latinos. You probably heard from my friend and colleague, Dr. Laura Fegerman, on the great benefits of studying these populations to identify new or common variation associated with the cancer, especially because some of these variants have an ancestry that is unique to the region, which is American Indian or Indigenous American. And this is an example of a paper led by Laura where we identified an Indigenous American gene that had a really strong protective effect for breast cancer in the region. So this is actually a unique finding that leverages the genetic ancestry of the populations to identify new forms of new genetic determinants of cancer. This is another table coming from a collaboration with then again with my friend Laura Fegerman and a larger group in California, Latin America, where we tested the effect of known SNPs for breast cancer, mostly discovered in Europeans in case control studies of Latino ancestry. And interestingly, we found that they actually performed rather well in the population, but we believe that more work needs to be done, especially because the American Indian component has been largely unexplored, and this is an effort that Laura is leading as part of the Confluence study. This figure shows the top 10 causes of cancer incidence and of cancer death in newer Latinos. And here on these red boxes, I'm showing the only cancer type for which there are polygenic risk score studies, so only breast cancer. So there is actually a big research gap in this population for cancer risk prediction, and we really need to identify more common variants that increase the risk of these other cancer types, as well as developing risk prediction models. So this is actually a big research gap that needs to be addressed at some point in the near future, I hope. I will now talk about tumor genomics. I will say that the most important limitations to advanced precision cancer medicine in Latinos is the lack of tumor genomic data. Tumor genomic data is important because we can use such data to identify mutations that are more common in the populations and that can be targeted with new therapies. So this table that you may have seen before, it shows, for instance, the CGA have a really small number of Latinos analyzed. So for instance, for prostate cancer, only 1 of 495 of the tumor sequences were from Latinos. So that means that we know very little about, what are the more common drivers in the population, and which therapies should be prioritized for development. My group is currently supported by the NCI to study genetic changes in gastric tumor from Latinos. This is a figure from a study that we have on the review, showing that gastric tumors in Latinos are highly heterogeneous depending on the molecular subtypes. There are 4 subtypes of gastric cancer. And we are interested here is in identifying not only what are the more common targets in the population, but also what are the targets that are clonal, i.e. that are present in all tumor cells that are more effective for therapeutic development. In this figure, I'm showing data for 4 molecular subtypes of gastric cancer from the previous slides. I'm showing here the fraction of tumors in this Latino population that can be potentially treated with targeted therapy. For instance, MSI, microsatellite stable tumors, nearly all of them can be treated with known compounds, target compounds, while genomically stable tumors, the fraction of them is actually lower. So this is actually the type of data we need in order to select the therapies that can be potentially more effective in the community. Tumor genomic data is also quite important to understand cancer etiology, and this is something else that we are doing in my group. This is data from the same study from gastric tumors for different mutation signatures, and we are actually starting to find very interesting aspects here. For instance, microsatellite stable tumor, the tobacco is quite important for clonal mutations or initiating mutations, while in MSI tumors is important for progression mutations or for non-clonal mutations. We are also seeing things like aflatoxins. We are seeing some effects of these known carcinogen for other cancer types in gastric cancer. So this tumor genomic data is not only important for targets, but also to help understand more about the etiology of these tumors in the region. In summary, there are many research gaps on the genetics of cancer in Latinos. I show you that the monogenic syndromes in the regions are quite interesting, and that we can leverage the high frequency of founder mutations to test new ways to use genetics to prevent cancer. With regards to common variants, clearly we need more geographic studies in the population and develop prediction models that are specific to these communities. And with regard to tumor genomics, we need more work again, because this is super important for us to optimize precision medicine and actually to understand more about the etiology of cancer using this signature analysis that I presented in the last slide. Finally, I would like to thank my group, my collaborators, and the NCI, particularly DCCPS, for the great support and for funding my study.
Great. Thank you very, very much, Luis, and great presentation. We'll take questions at the end. So our next presenter is Dr. Morgan.
Good afternoon. Is the volume reasonable?
Yes.
Can you hear me okay? Emil, can you hear me okay?
Yes, I think it's fine. Thank
Good.
you.
All right. Well, thank you. I'd like to thank Tram and Joanne and Shobha for their invitation this afternoon, and really congratulations for organizing this important symposium. I'd like to start with several contextual slides with respect to our program in gastric cancer epidemiology, and then focus on 3 works in progress, which are related to genetics, H. pylori, and environmental influences. The Central American 4 region is the core LMIC region in the Western Hemisphere, with a population of 40 million and at least 5 million immigrants in the US. The other LMICs are Bolivia, Paraguay, and Haiti. The CA4 region is united by their history, politics, culture, language, and poverty. And it is a geopolitical region with open borders as of 2006. The leading cancers are gastric, cervical, and hepatocellular, which to a certain extent mirror the Hispanic reality. And in these high incidence areas, we focus on prevention as well as accelerated research opportunities. The Western Honduras Gastric Cancer Program, with my counterpart, Dr. Ricardo Dominguez, and teams on the ground, has as its core an ongoing case control population-based gastric cancer epidemiology initiative, which has expanded over the years geographically and scientifically. We have 2 chemoprevention trials for patients with pre-malignant lesions, which is they are entering the analysis phase, funded by DCP and in collaboration with Dr. Cruz Correa. The cancer control initiatives focused on registries are primarily funded by NCI Global Health. And again, I'd like to thank my collaborators on the ground and in the US, particularly the EMSH lab at UNC, the Williams lab at Case Western, and the Peake and Wilson labs at Vanderbilt, as well as intramural collaborators. Three comments on gastric cancer epidemiology. First and foremost, it represents a marked cancer disparity in the US. The incidence rates among minorities, including Latinos, are nearly double among all non-whites. We have much work to do in this regard. Gastric cancer is the number 3 cancer globally, has significant geographic variability. The African enigma, the Asian enigma, but in Latin America, it is the altitude enigma. The burden of disease is primarily in the mountainous areas from southern Mexico to Chile, a biomarker to a certain extent. But it's not altitude per se, but this probably represents a surrogate for the aggregation of risk factors in the region. And this plays out with a match of the topography and incidence and mortality at the national level, as here is visualized in Colombia, and also at the sub-regional level. And then immigration matters. In the systematic review and meta-analysis, there was a remarkable consistency in the global literature as follows. Those who immigrate from high incidence areas to low incidence areas such as Western Europe and North America maintain the risk of their home country, essentially importing their H. pylori genetics and their host genetics. What does this look like in the US? Out of the 43 million foreign-born immigrants, there are probably about 13 million, or 1/3, that are at higher risk for gastric cancer. And there are really 4 regions with large populations in the US from high incidence areas. And these include Mesoamerica, the Andes, Eastern Asia, and Eastern Europe. And it's important to note that there are really 2 Mexicos with respect to incidence. The higher incidence areas in Mexico are really in southern Mexico. This is general observation. There are exceptions in areas contiguous with Central America. The Correa cascade, really diagramming the progression from gastritis to the pre-malignant lesions to cancer, is analogous to our other GI luminal cancers and offers points for intervention, and also raises the questions as to whether there are differential risk factors in the pre-malignant lesions, and particularly in progression to cancer among Latinos. I will comment, reflecting on Dr. Stern's comment this morning, that our societies, both European and US, are beginning to gauge gastric cancer risk and call for surveillance in immigrant populations and minority populations. So we're making progress on that front. The Bermuda Triangle of etiologic factors is a reasonable construct to consider differential risk factors among Latinos. Very simplistically, this is a decades-long gene-gene conversation between H. pylori genetics and responses and host genetics and responses, which is modulated by diet and environmental factors. I'd like to highlight 3 studies from these 3 domains in the remainder of my talk. We first asked the question using geospatial analytics as to whether the subtypes of gastric cancer may cluster. Here, a mapping in Western Honduras of the cases. In the upper part of the figure, the Caribbean, Belize, and Guatemala, and in the lower left, El Salvador. We did not see a clustering of intestinal gastric cancers, and this makes sense. This is primarily H. pylori driven with an inflammatory carcinogenesis pathway. We did identify a significant diffuse gastric cancer cluster as visualized on the right, which was highly significant by two different methodologies. And this group really had a proportion of younger patients with limited survival. This may well suggest germline or founder mutations and or ongoing endogamy. And whether CDH1, other known mutations, or new mutations are present is a work in progress. But I think this finding in the CA4 may have implications certainly for the region, if not for Hispanics in the US. H. pylori and co-evolution. Now, H. pylori co-evolved with the human species, and one can trace human migrations by looking at clonal groups. The co-evolution hypothesis, and this probably applies to other microbes, postulates that where there is a match of genetic ancestries, H. pylori is probably a neutral or beneficial commensal. Where there is disruption of coevolution or mismatching, that opens doors for disease. The Correa group looked at pre-malignant lesions in two different groups of subjects with genetic diversity, low incidence Afro-Caribbeans on the coast and high incidence Andean populations. What was unique about the study is looking at genetic ancestry on both sides of the coin. Each column here, moving from the upper panels to the lower panels, represents genetic ancestry, both on the host side and the H. pylori side. While the study was not powered for all permutations, what they did find is that premalignant lesions were only identified in Amerindians with African strains, which is intriguing. And the mathematical model emphasizes this with increased, which is based on the data, showing increased disruption or mismatching of genetic ancestry, moving from the lower left to the upper right-hand corner. We are now involved in a broader study currently underway, 3 sites in Central America. And I'll note that Central America and the Caribbean really have the greatest H. pylori and host genetic diversity on the planet. Initial results in our patients in the Copan region on the human side are as expected. Essentially Amerindians with 50 to 100% admixture, and with expected admixture with European populations. Now, on the H. pylori side, the phylogenetic analysis underscores the unique ecology of each group and potentially differential risk factors in Hispanic populations. You see Honduras in blue and brown here against the backdrop of 1,200 reference strains. And indeed, as you see on the left, we have 2 core groups identified, African on the left and Iberian on the right. And indeed, the initial analysis does show a significantly increased risk of cancer and premalignant lesions among Amerindians with African strains. So this is consistent with the coevolution hypothesis, and we look forward to expanding our sample size and diversity. Quickly to conclude on the environmental influences that are important, and we often forget about, at least those of us in GI, as we focus on H. pylori and genetics. But wood stove food preparation is linked to aerodigestive cancers and mediated by ingested and inhaled carcinogens, primarily heterocyclic amines and polycyclic aromatic hydrocarbons. We investigated the association between gastric cancer and H. pylori and wood stove use with modification by carcinogen metabolizing genotypes that characterize internal dose. And quickly, there were 3 primary findings. Both age, sex, wood stove use, and CAG-A were strongly associated with gastric cancer. 2 specific SNPs were also associated But importantly, as we see in column 4 and in this graphic, there was in our final model an important interaction between the CYP1B1 SNP and wood stove use. Remarkably, this interaction was independent but of similar magnitude as the highly associated CAG-A gene. And this interaction between wood stove use and genetics, both highly prevalent in this region, really informs us about who may be most at risk and reminds us that diet and environmental factors are important in gauging risk in Hispanic populations, and that the degree of acculturation may contribute to risk heterogeneity. So thank you very much. Gastric cancer is indeed positioned intriguingly in global health, immigrant health, and Hispanic health, and an interesting biologic Bermuda Triangle with potentially multiple differential risk factors among Hispanics, dominant and important in Latin America, and again in the US, a striking US cancer disparity. So thank you very much. I appreciate the opportunity to share some ideas.
Oh, that's great, Dr. Morgan. Really appreciate your thoughtful review of where we are with gastric cancer and Latinos here and internationally as well. And my presentation is going to, if I can have control now, we'll focus on liver cancer. My presentation will focus on liver cancer in the US, as well as how Latinos and Hispanics fare with regards to liver cancer. And I really appreciate the opportunity to look at this cancer, because overall liver cancer is kind of increasing for our population. And we really need to do some serious research in this area, because it's truly a life-altering disease that warrants our attention and efforts to help resolve it. So I'll quickly review some of the data that we have for Latinos in the US, and then what we're seeing here in South Texas and some of our own research, and then ideas for potential research in the future. We all know that there's different forms of liver cancer, but what we're going to really focus on is on primary liver cancer, which is referred to as hepatocellular carcinoma. And what we find that this is the most common type of liver cancer, and of those that are impacted by it, it affects 75 to 80% of the individuals. And the sad thing about it is that approximately 70% of liver cancer cases in the US could potentially be prevented through the elimination of various risk factors that are listed on this slide, such as excess body weight, type 2 diabetes, chronic infection of HBV or HCV, and heavy alcohol consumption and tobacco smoking. But risk is also increased by eating certain contaminated foods by aflatoxin. However, we need more research in this area. And aflatoxin is a poison from a fungus that grows improperly in stored foods such as nuts and grains. And then interesting, we are having some accumulating evidence that suggests that drinking coffee may reduce the risk of liver cancer. But again, we need to focus more on the differences of racial and ethnic risk factors for this disease. Many of you have seen this slide of the top 10 types of cancer. On the left here, you will see the incidence of the top 10 cancers among Hispanic males and females in the United States, where liver cancer is the 8th most frequent cause of cancer for Hispanics. And on the right, you'll see the cancer mortality, and liver cancer is the 5th most deadly cancer for Hispanics or Latinos. And I believe Luis showed this graph as well, but when we look at the geographic distribution of new cases among Hispanics in the United States, we can see, as expected, those are occurring in states like Texas, New Mexico, and Arizona with large Latino populations. But when you look at the absolute numbers, not only Texas, but California, Florida, and New York appear to be important states regarding the liver cancer burden, and again, states with large Hispanic populations. Mortality patterns are similar to case patterns from the previous slides. And here we can see mortality numbers are adjusted rates due to liver cancer. They're high in Texas and California and also in Florida. And we'll notice that California has more death-related deaths due to liver cancer. However, when you look at the adjusted liver cancer death rates, it's actually higher in Texas. And when you look at South Texas, I will be showing you that we have even higher rates of liver cancer in South Texas. And this happens to be the region of our cancer center. So now just quickly reviewing what we're finding here in South Texas, even though our South Texas population is majority are Hispanics, Mexican Americans at 69%, nearly half speak Spanish as their primary language, and about a quarter live in poverty, and about a quarter have not completed high school, and per capita income is low at $30,000. But in this kind of environment, it's not a surprise to see the cancer disparities for South Texas that are noted here in gray, and South Texas Latinos that are noted in blue compared to the rest of Texas and the nation. And the dark bar represents the US numbers. And as you can see on your right-hand side, liver cancer really takes a large jump in our South Texas population. And in this slide, we're looking at liver cancer in Texas from 95 to 2015, utilizing the standardized incidence ratio. And again, as you can look here at South Texas, you can see the darker blue counties represented with the highest number of liver cancer incidence rates. And now what some of our research reveals that in our study back in 2012, we published one of the first papers describing the epidemiology of liver cancer in Texas and comparing Texas to South Texas and to the US SEER population. And again, I interchange between Hispanics and Latinos based on some of the work that we're doing here. On that first paper here on the left, this is the time span that you see for South Texas and Texas Latino cancer liver rates that are rising at a much higher level than we see for the US population. And on the right, you see the South Texas and Texas Hispanic liver cancer rates increasing at younger ages and holding steady at the older ages, while the US Hispanic rates begin to decrease at age 75. We also did some additional research to find out what were some of the things that were contributing, or what were the risk factors associated with this increase. And we found that obesity increased in all three populations of Hispanics, with the highest increase among Hispanics in South Texas. And diabetes also significantly increased among US Hispanics. And there was also a non-significant increase in Texas and South Texas Hispanics. We also looked at alcohol use and did not see any change significantly over time among the three Hispanic groups, though there were non-significant increases in South Texas. And in terms of cigarette smoking, we see significantly declined only among US Hispanics between the time period of 2004 and 2006, and there were no significant differences among our three population groups. Then in 2014, we updated our numbers for our second research paper looking at the incidence of liver cancer. And we found similar trends as our previous analysis, where South Texas and Texas Latino liver cancer rates increased at a greater level than US Latinos. And in this slide, we also show that in terms of the age groups, if you'll take a close look at the top left here, you'll see that the age 50 to 59 is still the group with increasing trends across all groups, but the rates among Hispanics for Texas and South Texas are increasing at a higher rate compared to the US SEER Hispanic data. In 2017, we published the results of a small case control study that was funded by the Cancer Prevention Research Institute of Texas. And what we found here was that significant differences between our cases and control. For cases reported more smoking and alcohol use and were more likely to have HCV positive and have aflatoxin in their blood and urine. They reported less use of aspirin, statins, and omega-3 use. And they also reported having less private health insurance. So this may be a confounding factor in some of our data. But risk is also increased by eating foods contaminated with the aflatoxins, which I just mentioned, that we are very interested in looking further and investigating this potential impact on liver cancer. And here are a few studies that are also looking at the aflatoxin hypothesis, where we have you et al. Also focused on East and West Texas, which also has a high risk of liver cancer. They found a shift in the aflatoxin contamination pattern in the state of Texas, which may warrant further studies on the risk assessment for the potential etiological effects of such increased exposure. And on the lower right, UT MD Anderson cancer researchers aimed to determine whether aflatoxin dietary exposure plays a role in the high incidence of HCC observed among Hispanics in South Texas. There they detected, there was a mutation was detected that 3 of 41 of the liver cancer tumors from Hispanics in South Texas. And then the mutation was detected only in the Hispanic and Asian liver cancer patients and patients harboring the TP53R249S mutations, which were significantly younger and had a shorter overall survival rate. And aflatoxin exposure may contribute to a small number of liver cancer cases in Hispanics in South Texas, but the detection of that same genetic marker mutation in the plasma cfDNA did not appear to be a promising biomarker of risk assessment for liver cancer in some subjects with cirrhosis or advanced fibrosis in this population. Another update is that we found consistently Hispanic males had the highest rates across all regions and data sources. Also, there's a clear gradient from the highest in South Texas to lower at the national level. Rates among Hispanics for males and females were anywhere between 2 to 2.8 times that of their non-Hispanic white counterparts across all regions and data sources. We found similar trends as our previous analysis. South Texas and Texas Hispanic liver cancer rates increasing at greater proportions than US Hispanics. Again, Hispanics had higher rates than non-Hispanic whites across all our regions and our data sources. And here in Texas, the Cancer Prevention Research Institute has issued a special opportunities to do liver cancer research. And so we're looking forward to these new potential outcomes of some of the studies that have been funded. And just to kind of conclude in terms of our final remarks, we can't ignore the impact that COVID-19 has had. And although this implies in general for all cancers, I believe it's a crucial importance for liver cancer as it shares several risk factors with COVID-19. While measures are diverting healthcare resources and suspending non-urgent care because of COVID where necessary, delays in cancer screening, diagnosis, and treatment due to reduced healthcare access will likely result in a short-term drop in cancer diagnosis, followed by an increase in late-stage diagnosis and preventable cancer deaths. So the American Cancer Society also alerts us on that fact, and that some individuals may continue to delay preventive care and symptom follow-up due to the fear of exposure or loss of employment or other employer-based healthcare issues. And that's going to have ramifications for us and will particularly affect historically disadvantaged communities that already have challenges in accessing quality medical care and face a disproportionate burden of COVID-19 illness, hospitalization, and death. And additionally, behavioral changes that have been adopted during the pandemic, such as weight gain, physical inactivity, and increased alcohol consumptions, may carry over into long-term health consequences that we need to pay attention to. But again, there's different types of HCC that we need to start thinking about as well. And a key priority in this area is the need for more research to identify determinants of the progression of non-alcoholic fatty liver disease, or NAFLD, to liver cancer, given the burden of this cancer among Hispanics. Plus, we need more research to address obesity among Hispanics, particularly in childhood issues, as we see more than a third of our Hispanic Latino children are either overweight or obese. We see that Hispanics are more likely to develop NAFLD and related HCC compared to non-Hispanic whites. And earlier studies have reported that Hispanics were more predisposed to NASH. And Alcatraz et al. Also found an increase of liver cancer among Hispanics. In addition to US-born Hispanics had a higher incidence of liver cancer when compared to foreign-born Hispanics, suggesting that, again, as we've been hearing from some of our presenters, that environmental, social, economic, and cultural differences may be contributing to these factors as well. So we need to work together. We know that NCI really wants us to help us identify the gaps and opportunities for additional cancer epidemiologic research in Latinos. And so we recommend that we need large prospective studies, including multi-level risk factors, biospecimens, as well as the standardization and implementation of critical data collection relevant to Hispanics and Latinos, at different levels, at the provider level, and other areas, to enrich our cancer registry data. And we also need to facilitate and coordinate across discipline collaborations to inform research in Hispanic populations. And that research should appropriately focus on looking at the risk factors that contribute to liver cancer, such as obesity and diabetes, and other risk factors that underlie this life-threatening disease. So thank you very much. And I'll take questions at the end of our panel. And our next presenter will be Dr. Adam DeSmith, who will be talking to us about childhood cancer health disparities in Hispanic populations.
Good afternoon, everyone. I'd like to thank the organizers for inviting me to speak at this very important workshop. I'll be speaking about childhood cancer health disparities in Hispanic populations. I would jump right in if I can find the arrow. Oh, here we go. Okay, so childhood cancer comprises a spectrum of different malignancies. The spectrum of different malignancies are distinct from the common adult tumors that we're all familiar with. So acute leukemias comprise the largest subgroup of childhood cancers, followed by tumors of the central nervous system, including pediatric brain tumors, followed by lymphoma and soft tissue sarcomas. And it is estimated that around 11,000 new cases of childhood cancer will be diagnosed in the US this year. Despite improvements in cure rates for certain childhood cancers over recent decades, cancer does remain the leading cause of death from disease among children. And unfortunately, children who do survive cancer can face lifelong treatment-related morbidities. In a study of SEER data in around 400,000 children, childhood cancer survivors, in the 5-year survivors among the 5 to 19-year-old age group, around 2/3 developed any chronic health condition. And this rose up to around 90% in the 40 to 49-year-old age group of childhood cancer survivors. And approximately 1/3 of childhood cancer survivors were found to have a severe, disabling, or life-threatening chronic condition. So there exist several childhood cancer disparities that affect Hispanic populations. Probably the most well-known one is the increased incidence of acute lymphoblastic leukemia among Hispanic children. So acute lymphoblastic leukemia, or ALL, is a cancer of white blood cells and is the most common childhood cancer. And children of Hispanic ethnicity have up to a 40% increased risk of ALL than non-Hispanic whites. And indeed, Hispanic children have the highest risk of ALL in the United States. But this increased ALL risk actually occurs across the age group in Hispanic individuals. And indeed, the adolescent and young adult group have around a twofold higher risk of ALL in Hispanics than non-Hispanic white AYAs. Intriguingly, ALL incidence has been found to be rising in recent decades, but this increasing incidence has only been found to be significant in the Hispanic population. The reasons for this increasing incidence of ALL in Hispanics is unknown, but clearly our genetics are not changing that much. So, this points towards changes in environmental exposures. In addition to the increased risk of ALL, Hispanic ALL patients have inferior survival. Back in 2003, this was a study from SEER data from 1973 to 1999, showing Hispanic ALL patients in the dotted line here had inferior survival compared with non-Hispanic white and Asian patients. And this disparity appears to persist into recent years. In this more recent data from 2005 to 2011, Hispanic childhood ALL patients had inferior overall survival and event-free survival than non-Hispanic patients. And there are likely several reasons for this disparity, including socioeconomic and environmental factors, differences in tumor biology and genetics, and including differences in genetic ancestry, which I'll touch on more later. Briefly, there is evidence that Hispanic children with increased Native American ancestry have inferior survival. So a recent study from Shoag et al. Found that continental Hispanic children had higher mortality than Caribbean Hispanic children, compared with non-Hispanic whites with ALL. I'll come back to ALL later, but I wanted to briefly talk about some disparities of other childhood cancers. In a recent study from Erin Marcotte and colleagues, where they studied childhood cancer incidence using SEER data from 2000 to 2017 and looking at single years of age, so looking at narrow time windows, they found that incidence of CNS and lymphomas were lower in Hispanic children, but ALL was higher, as we know. And in addition, there was increased incidence of retinoblastoma, hepatoblastoma, and osteosarcoma. Overall, there was a decreased incidence of Hodgkin lymphoma and gonadal germ cell tumors. But interestingly, the incidence or risk of these cancers appears to be higher in the younger age groups of children, as shown in these figures, where you see this bump in the incidence of ALL among the younger Hispanic age group for both Hodgkin lymphoma and gonadal germ cell tumors. And in terms of outcomes, Lindenberry and Ross reported in 2008 that Hispanic children have a poorer 5-year survival than non-Hispanic white children overall. And in overall cancers, and in terms of specific cancers, including leukemias, CNS tumors, Ewing sarcoma, and melanoma. More recently, Green and colleagues reported that Hispanic children with leukemia or central nervous system tumors have a greater risk of early death than non-Hispanic white children. As I've touched upon, socioeconomic status likely contributes to this disparity. And it was indeed recently shown that SES partially mediates the high risk of mortality in Hispanic children for ALL, AML, neuroblastoma, and non-Hodgkin lymphoma. Because this was found to only partially mediate, this suggests that there are additional factors that underlie this disparity in survival. For the rest of the talk, I'm going to focus on potential causes of the increased risk of ALL in Hispanic children. We recently published a review article, and this is a figure from that, where we show risk allele frequency among among the established GWAS loci for acute lymphoblastic leukemia. And we're showing here the risk allele frequency in Latino admixed American populations versus European populations from the Genome Aggregation Database, or GNOMAD. And for several of the well-replicated GWAS hits, we found, we showed that there's a higher risk allele frequency in Latino populations. And in a recent transethnic meta-analysis GWAS of ALL, using California-based ALL cases and controls, we constructed polygenic risk scores using the known GWAS loci. And we showed that among all individuals, there was a higher polygenic risk score in Hispanic individuals compared with non-Hispanic whites, which was highly significant. And when we stratified by case control status, we found that even among controls, there was a significantly higher polygenic risk score among Hispanic controls compared with non-Hispanic white controls. So we think that this shift towards a higher polygenic risk score may partly explain the higher incidence of ALL in Hispanic children. In two recent large GWAS of ALL in Hispanic children specifically, these discovered a novel risk locus at the chromosome 21 gene ERG, which showed stronger effects on ALL risk in Hispanic than in non-Hispanic white children. And when looking at genetic ancestry, the ERG locus, among Hispanic children with higher Native American ancestry, the ERG locus had a stronger effect on ALL risk. And this was the case both looking at genome-wide Native American ancestry, as well as looking at local Native American ancestry. So for example, in Hispanics with at least one copy of the Native American haplotype, there was a stronger effect of this ERG risk locus on ALL than when looking in Hispanic individuals with 0 copies of the Native American haplotype. And we think that larger GWAS of ALL in Hispanic populations may reveal additional risk loci and help us to improve risk prediction using polygenic risk scores in this population. Previous GWAS of ALL in Hispanics have also identified that risk alleles in several of the known GWAS loci are associated with increased Native American ancestry. For example, the ARID5B risk SNP has been associated with Native American ancestry and also associated with patient relapse. And Native American ancestry itself has also been associated with patient relapse, with higher Native American ancestry conferring a greater risk of relapse. The GATA3 risk SNP, the risk allele frequency of this has been found to be over 2 times higher in Hispanics than in non-Hispanic whites. The GATA3 risk SNP has been specifically, or at least much more strongly associated with the pH-like molecular subgroup of ALL. And we know that the pH-like subgroup is more common in Hispanic ALL patients and is associated with poorer outcomes. So it is likely that the GATA3 risk SNP contributes towards the inferior survival of Hispanic ALL patients. Briefly moving away from genetics, so there's likely environmental risk factors that also contribute to the increased ALL risk in Hispanics. The delayed infection hypothesis of ALL etiology proposes that infants or children who in early life are unexposed or have low exposure to infections, who are then exposed to infections in later childhood, will have an overreactive immune response, leading to increased risk of leukemia. And there have been several epidemiological studies that have studied this. And for example, daycare and higher birth order, which are both for proxies for infectious exposure, have been found to be protective in non-Hispanic white children, but these were not found to be protective in Hispanics, suggesting differences in the etiology of disease between populations. In contrast, cytomegalovirus infection at birth, which has been shown to be a risk factor for ALL, was shown to have a stronger effect among Hispanic children than non-Hispanic whites. This was in the California Childhood Leukemia study. And also cesarean section, another risk factor for ALL, again, was shown to have a stronger effect on ALL risk in Hispanics. We think that cesarean section may impact ALL risk perhaps through impacting the fetal microbiome and neonatal microbiome. And indeed, a study in Nature a couple of years ago showed that there were distinct differences in fetal microbiome that persisted several weeks into infancy when looking at vaginal births versus cesarean section births. And intriguingly, it's previously been shown that there are racial and ethnic differences in microbiome, in vaginal microbiome, as you can see here in this figure. And of course, there may be gene by environment differences between populations that may also contribute to the increased risk of ALL in Hispanics. Just some outstanding questions. How much of the Hispanic ALL risk is due to genetic variation versus environmental exposures? What factors may underlie the rising incidence of ALL in Hispanic populations? And how can we reduce ALL risk in predisposed individuals? And finally, how can we address the existing childhood cancer survival disparities in minority populations? And I think that a lot of these questions can also be applied to the adolescent and young adult population. Thank you.
Thank you very much, Adam. A great presentation, and so much that we need to learn in terms of ALL in our Hispanic children, as well as their young adults. Our next presenter is going to be Dr. Frank Benedo from the University of Miami, discussing cancer survivorship in Hispanics.
Good afternoon, everyone. I'd like to thank the NCI and the organizers for inviting me to talk about cancer survivorship in Hispanics. I have to say this will be a little bit of a shift in focus, as I'm going to be talking more about the psychosocial and quality of life experience of Hispanic cancer survivors. Well, in any case, let me begin by disclosing I'm a paid consultant for Blue Note Therapeutics, which is digitizing some of the intervention work that I'll be presenting today. So today I'm going to be talking about some general concepts about cancer survivorship, and particularly some of the challenges that are faced in the Hispanic community, particularly in what we know about determinants of optimal adjustment to cancer diagnosis and treatment. Then I'm going to very briefly touch upon two studies where we're targeting these Hispanic issues and populations to either learn more about mechanisms that impact outcomes or to improve quality of life in these communities. And I'll wrap it up with some concluding remarks. I know many of you have seen the slide, but currently there's about 20 million survivors in the United States. That number is expected to reach about 26 million in 2040. The vast majority of our survivors are 65 and older, which raises some challenges in terms of comorbidities that exist with the chronic and debilitating symptoms that are a consequence of many treatments for cancer. If we look at psychosocial challenges across the cancer survivorship continuum, from diagnosis to long-term outcomes and the end of life, our survivors experience, rightfully so, at each pivotal point in the continuum, several psychosocial stressors and emotional and psychosocial reactions that can be challenging. So for example, uncertainty and anxiety when one is diagnosed, issues related to treatment planning with information overload and having to make decisions, particularly now in the era of complex available treatments. Once treatment begins, of course, the symptom burden can lead to interpersonal disruptions. There's concern in monitoring over the treatment response, and cost can be also a major stressful factor for survivors. Down the continuum, we continue to see late effects of treatment well beyond the primary treatment period, and of course, issues specific to surveillance and recurrence persist. Now, we know there's a pretty robust literature showing what factors may contribute or exacerbate these challenges and lead to poor quality of life. And as you would expect, these tend to be older age, more comorbidities, poorer pre-morbid functioning, having to navigate complex health systems, limited access, poor health literacy, and information and financial burden. We know that by 2030, there's going to be 4 million cancer survivors in the United States of Hispanic Latino origin. That's going to account for 20% of all survivors. And we know that the Hispanic community as a whole has a disadvantage regarding socioeconomic status and access to equitable treatment, as well as low rates of health insurance and financial resources. Something that's also very troubling is that even when you have proper access and proper SES, Hispanics in general continue to experience disparate treatment options, including low inclusion in clinical trials or limited access to novel and experimental therapies. We have documented and discussed the challenge that these disparities can create for Latino survivors, particularly how the fact that they tend to be poorer, have lower education or no health insurance or source of primary care relative to non-Hispanic whites can contribute to some lower survival rates, even after accounting for differences in age and stage distribution. So these social determinants are key players in impacting not only the physical health, but also the quality of life of our Latino survivors. Work has also been focused on understanding the burden or the psychosocial burden of survivorship on patients in terms of quality of life. For example, we know that breast cancer, colorectal cancer, and prostate cancer survivals that are of Latino background tend to report poor general quality of life, poor mental, physical, and social quality of life. And in some cases, in the prostate cancer literature, we find that they also report poor disease-specific quality of life, like sexual functioning. In a recent review by Samuel, she looked at multiple studies, and consistently she found that Latinos and Hispanics were reporting lower quality of life and higher symptom burden relative to non-Hispanic whites, and also more suboptimal physical well-being and lower social well-being. In contrast, there were higher rates found of spiritual well-being and spirituality-based coping in the Latino community, which has some implications for how we work with that community. So if we think of the Hispanic population as having these social determinants that could present some challenges ranging from access to care, limited knowledge and health literacy, which influence late presentation, disease severity, and also the context of having greater comorbid diseases was mentioned earlier, particularly rates of diabetes and obesity and other conditions that require management while having to manage cancer. The question for us becomes, do these determinants exacerbate or negatively impact the psychosocial experience of cancer survivors? And although we're not going to be able to dive into considered survivorship care itself, do they also impact prevention, surveillance, intervention, and coordination of care post primary treatment for the survivors? Some work has shown that Hispanics may be at a psychosocial disadvantage. A lot of this work actually comes from the cardiovascular literature, but there is documented studies that show that Hispanics tend to have greater levels of perceived stress, less knowledge and controllability over their illness. They tend to be less assertive, particularly when dealing with the health system, tend to have more fatalistic attitudes and less optimism, engage in greater avoidant coping, and rely more on the family, which is not a bad thing if you have a cohesive family, but if you're isolated from your family or you have a conflicted family, that tends to be problematic. And we also know that Hispanics tend to be less motivated to adopt behavioral changes unless they see a benefit for the entire family or for the group. So the question is, do these patterns render a psychosocial disadvantage to the Hispanic cancer survivors? So we know that cancer diagnosis and treatment can impact quality of life. We also have a very robust literature showing what are the mechanisms that may buffer the stress associated with cancer and its treatment and treatment-related side effects, and how these may prevent deteriorations in quality of life and other outcomes. If Hispanics do have deficits in these particular mechanisms like controllability, knowledge, emotional expression, and may it sometimes have isolation from their families, does that put them at greater risk of having decrements in their health-related quality of life? So we've done some work to try to understand what are the unmet needs of this population. These are some findings in a sample of 288 breast, colorectal, and prostate cancer survivors that were studied in Texas and Illinois. As you can see, most of them, or half of them, had an income of less than $25,000. Most were foreign born, and most were monolingual Spanish speaking. And what we found was that patients in the sample, or survivors in the sample, had high rates of unmet needs, ranging across psychological, health system and information, care and support, physical, and sexuality. And that many of these needs were associated with less satisfaction with care and with greater symptom burden, both in prostate and in breast cancer. These are in the negative direction, as lower scores suggest a better quality of life. So in colorectal cancer, our instrument didn't hold well psychometrically, so we were not able to test that, but we did find a consistent pattern for the most part, finding that these unmet supportive care needs in survivors were associated with symptom burden. When we dissected to see what were the issues that our patient population was raising, fears of recurrence was the main concern stated by the sample. For prostate, sexual functioning; for breast, fear of recurrence, and for colorectal survivors, concern about being a burden on others. What's interesting here is that the two major concerns were in the psychological domain, again, fear of recurrence and concerns for close others. And what we saw was when we compare these rates to non-Hispanic whites, we find the same patterns, the same classifications of what the concerns are, but we do find higher rates of unmet needs in the Latino samples. This is just maybe a deeper look at self-efficacy. Self-efficacy is a key construct in one's ability to believe or perceive that they can manage and take care of their health condition, and that if they do so, that they would have a favorable outcome. So we have found that, for example, satisfaction with care, which is a strong predictor of health-related quality of life, is robustly associated. But we have also identified that that association is mediated by self-efficacy. So it's not only about being satisfied with one's care, but also believing that they have controllability and that they can make change happen in regard to various domains, ranging from communication through psychosocial factors, social support, and social and recreational activities. And of course, these models control for acculturation, nativity, and other clinical covariates. So I'd like to just briefly mention two studies that were conducted now. One is an intervention study, another is a cohort study. In the intervention study, which is Encuentros de Salud, we're evaluating the incremental efficacy of a culturally adapted cognitive behavioral stress management intervention for Hispanic prostate cancer survivors. As you know, prostate cancer is the most common non-skin cancer diagnosed in Latino men, and it is the fourth leading cause of death. And what we're testing is a model. This is some prior data where we had shown that Hispanics consistently reported disadvantage in regard to optimism, ability to express emotions, and tangible support. And if we look at quality of life, we also find that Hispanic prostate survivors have lower rates of general social, emotional, and functional well-being. So what we did is we decided to test the intervention versus an adapted form of the intervention, where we looked at the literature and selected specific cultural factors known to implicate the therapeutic process. So familism being one of them, which is a strong attachment and interdependence with the nuclear family, which has implications with the individual to gain benefits or engage in a psychotherapeutic program. They have to see benefits to the extended family network. And we did that for simpatia, for other cultural factors like fatalism and male gender roles, and embedded these concepts into the delivery of the intervention itself. We didn't change the cognitive behavioral skills that were provided in the intervention, which are listed here, these are very straightforward techniques that are used in many evidence-based treatments. But what we did is we augmented the discussion about fatalistic attitudes and those distortions, the importance of self-efficacy. We played a lot more emphasis on social relations and the family, and also on assertiveness training. This is the intervention model. It occurs over 10 weeks, about 1 hour a week. It's got the cognitive behavioral component, which I briefly went over, and then several relaxation techniques. And the idea is that men participating in these groups are going to be able to improve their quality of life and reduce symptom burden through improvements in neuroimmune regulation and psychosocial and behavioral factors. And then the question for us is, can we find incremental efficacy in this intervention if we deliver it as a culturally adapted program? And we're in the midst of testing that intervention right now with 260 Latino prostate cancer survivors. I'd like to wrap it up by just a brief, and I don't know how I got back to my first slide. Oops. Apologies about that. I have no idea how I went back, but I just wanted to show a couple of brief slides on a recently funded study that I have the privilege of co-leading with Dr. Ramirez. It's Avanzando Caminos. It is the Hispanic Latino cancer survivorship study, and it's a study that's taking place both in South Florida and Southwest Texas. This study has several aims, which include establishing a cohort of 3,000 Latinos examining multiple determinants of symptom burden, quality of life, and disease activity, and then test multiple pathways that may impact the proposed outcomes. The cohort will recruit 3,000 participants. 50% will be Mexican American, 50% other Hispanic, 50% female, 70% will come from our cancer center, about a third from our registry, and a third will be rural patients. The model is a pretty straightforward model, which is postulating that symptom burden, quality of life, and disease activity are driven by various domains. Here we have two key domains as major predictors, which are socioeconomic and cultural factors and stress and adversity domains. And then we have lifestyle and behavioral factors, psychosocial and biological mechanisms that may mediate or moderate associations with the study outcomes, along with the typical demographic and medical factors. And in terms of biology, we're looking at leukocyte gene expression, cardiometabolic markers, and genetic admixture. We're recruiting, like I said, 3,000 participants in South Florida and Southwest Texas. Our participants and the numbers that we're looking at are guided by representation that we see in the Texas Cancer Registry and then the Florida Cancer Data System to be representative of what we see also in the community for our Latino participants. So I'd like to wrap it up by just saying that there's strong evidence that Hispanic survivors are at a disadvantage specific to social determinants, possibly due to some sociocultural factors. We do have a significant amount of heterogeneity, and I think looking at mixed samples of Latinos is going to help us get at some of that variability. If it does exist and it's significant, we'll be able to identify it. I'd like to again reiterate that these are complex models. These are not linear interactions. Familism and family interdependence, it's not good always. It depends on the context of the family system. One thing that I always worry is about stereotyping our participants by thinking that all Hispanics are fatalistic or all are family interdependent. We know that's not the case. And thinking of additional work needed, I feel that more work is needed looking at care delivery and the more prevalent cancers in Latinos as we're looking at a more diverse Hispanic groups. And finally, it's not all negative. I didn't have time to get into it, but we also look at resiliency factors that may buffer the negative impact of cancer diagnosis and stress. So with that, I'd like to acknowledge my group of collaborators across these multiple institutions. And again, thank you for your time.
Great. Thank you so much, Franco. A great presentation. We have a few minutes for Q&A, and I'd like the panelists to turn on their videos so that we can see you. And we have one question that I'd like to start off with, but please, the audience, please submit your questions. The first one will be for Dr. Carvajal or Dr. Strong. What is the prevalence of H. pylori by racial and ethnic groups in the United States?
I can have a go at that. There is data from a study, I think was in the VA administration, that was actually posted on the link. I mean, I don't have the specific number, but clearly there is a strong correlation between as Douglas showed, where you come from. So there is a higher prevalence in immigrant populations, especially if they come from the Andean region of South America, from Mesoamerica, which is Guatemala, El Salvador, and Honduras, and from certain parts of Asia. But Douglas can complement on that, actually.
Dr. Strong, would you like to add to that? Dr. Morgan, I'm sorry.
Sure. Can you hear me?
Yes.
Yeah, good. No, very important question. Overall, H. pylori is the most common chronic bacterial infection in the world. About half of the world is infected. As gastric cancer, it is an infection primarily of lower socioeconomics, both in the US and in Latin America. In the US, among minorities, in the VA study and the prior, which is now an older CDC study, for Hispanics and African Americans, it's around 50%, but dropping rapidly among young Caucasians, particularly middle class and above. So very intriguing phenomenon. In our work in Central America, it is the seroprevalence is on the order of 90%.
Thank you both very much. Elena Martinez, you had a comment. Would you like to verbally share that with the group? She can't unmute.
Someone has to unmute her actually, but I think while someone helps Elena, I will say that I think something that is missing from most of these studies is that not all H. pylori are equal. There are some strains that are more aggressive and that have a higher risk of cancer. So that is data that is still missing in the US, I would say.
Great. And also, if anybody knows of Ed, would you like to comment? Ed Trapito just placed a comment. Trying to encourage more interaction here. Okay. Well, I see Manuel Espinosa has his hand up. Can we unmute Manuel?
Hello, thanks to you, Dr. Ramirez. So I had two questions actually. One is for Dr. Carvajal and Dr. Morgan. It's as you mentioned in one of your slides, Dr. Morgan, there's clusters of gastric cancer endemicity in the world that travel along with migrant populations. Something that we've understand from, I'm a Hemong fellow, by the way. Something that we understand in the clinical oncology world is that gastric cancer in East Asian populations behaves differently clinically than gastric cancer in European populations. It responds better to therapy, and they actually use different chemotherapy agents in Japan specifically to treat that are similar to the ones that we treat here, that we use here, but they're not the same. So based off on that, I know that there's some like ancestry data linked because of how human migration patterns came from like East Asia and Siberia to North America and then to South America. A lot of indigenous and Native American peoples have some East Asian markers. So I don't know if you had any data on how the behavior or the genetic signature of this gastric cancers, where they're H. pylori induced or not H. pylori induced, if they're similar to the East Asian kind. So that's my question for both Dr. Carvajal and Dr. Morgan. And then for Dr. D. Smith, I have a question. I've seen ALL, particularly in Hispanic children and young adults. And I know that you mentioned, the conflict or the possibility of CMV in uro infections and the different genetic signature. What I had a question on is there also I've seen some higher prevalences of other blood cancers that are not as common, like T-cell lymphomas, EBV, HTLV1 based lymphomas, NK cell lymphomas. I don't know if you had any data. And again, this is also another type of lymphoma that we also see in East Asia a lot, or higher than in European populations. So I don't know if you had any data on those like T-cell, NK cell lymphomas, and whether or not they're also linked to ancestry. Thank you.
I may have a go at the first question. Actually, I'm not clinician. I'm a humble PhD, but I can tell you that there is a difference in molecular subtypes between populations. We have some data on that. In Latinos, we have a high prevalence of the most aggressive molecular subtype of gastric cancer, which is called genomically stable. These particular types tend to have diffuse histology, and they do not respond to standard chemotherapies. Okay. Other populations have a higher frequency of more responsive tumor types, such as those with microsatellite instability or Epstein-Barr virus infections that respond better to therapy and are also amenable for novel immunotherapy. So gastric cancer is being redefined by these molecular subtypes, and they may explain why we have differences in responses across populations.
Great. Dr. Morgan, do you want to add to that?
Well, I'd echo that, and Luis may be able to add input, but that reflects on really the success of the TCGA initiative with respect to gastric cancer. There was consistency among the different omics platform that really underscored the 4 molecular subtypes. And that's provided us with, not us, I'm not an oncologist, but provided us with actionable targets. So as Luis was referring to, what will be interesting in terms of East versus West is whether there's a difference in responses to these actionable targets. I know there's excitement among GI oncologists in 2021 with respect to gastric cancer because the emergence of a number of novel therapies is noticeable this year. And so that's exciting. And it probably does grow out of the TCGA.
Great, thank you. Then there was a question for Dr. DeSmith.
Yeah, I should give a disclaimer that my research is really focused mainly on childhood ALL. But yeah, as far as I'm aware, I haven't come across reports of other hematologic malignancies being significantly higher in the Hispanic population. I've heard some anecdotal reports about familial heme malignancies being enriched in Hispanic Latino families, but yeah, I wouldn't wish to comment any more on that question.
And are there any new therapies being developed for Hispanic children with ALL? Because we have high rates of our children in South Texas that have ALL, but we understand the treatments are not as effective for them.
Again, I'm an epidemiologist, not an MD.
Okay.
But
Okay.
I know there are studies looking into that. For example, there's Jun Yang, who's done a lot of research on pharmacogenomics, has identified, for example, variants that are specific to Hispanic populations, as well as different ones in Asian populations. So hopefully the field will move more towards targeted therapies because the current regimes, still are using the same chemotherapy drugs that were used 30, 40 years ago. And although we're curing these children, they're getting these lifelong chronic health conditions that, it would be great if we could reduce these.
Great, thank you. Frank, we have a couple of questions for you. One, can you expand on the positive resiliency factors? And then I have another question for you, and then I'm going to go to Elena, and then I'm going to go to Ed.
Sure, I didn't get a chance to talk much about that, but we do have resiliency factors in the Hispanic community. Again, the family being one of them. Many of our participants do have extended families. They're highly family interdependent, and they have multiple sources of both emotional and tangible support that do help them get through the cancer experience. Where we tend to run into challenges is when we have immigrants that may be isolated here in the United States, who may have their families back at home and they're here working hard, providing for them. And we know from some of the literature that Hispanics tend to seek support from their family before they would go to their friends or coworkers or other sources. And that's when we can run into a wall. So we work with our participants, particularly if they're isolated, on helping them facilitate approaching other sources of support where they may have to disclose the cancer diagnosis and the challenges that they're suffering, which is, it's hard for them given the remaining stigma that cancer carries for many Latinos.
Great. And another question for you was, how do you develop interventions for Hispanic Latino survivors that are in tune with the cultural heterogeneity of the group?
Yeah, that's a great question. So we're targeting Hispanic monolinguals right now, which by default are less acculturated and may have more prevalent patterns that we try to target through the intervention. In terms of heterogeneity from Hispanic background, I have to agree with Eliseo. I've been doing this for 20 years now, and being at Miami, we mostly see Cuban Americans, but 40% of our samples tend to be Central South American, and the heterogeneity that we see in country of origin really doesn't impact a lot of these factors, are typically explained by socioeconomic status and education to some extent.
Great. And for Dr. Elena Martinez, I think you had a question with regarding to H. pylori or wanted to provide a comment.
Oh, yes. Hi. Thank you. I finally got unmuted. No, I wanted to advocate for more recent population-based data on H. pylori infection. As I put in the chat, we have data from the VA, from the Southern Community Cohort, from the SOUL study, but these are not population-based studies. So I don't know if you knew, unless I'm mistaken, the last NHANES survey that provided data on H. pylori infection was from 1999. Those are the latest data that we have that are population-based. So I really wanted to advocate for getting more recent data on the various racial ethnic groups and this infection.
Great, thank you very much. Good to hear you, Elena. Ed Torpedo, Dr. Torpedo?
Yeah, hi. This is actually a question for you on liver cancer. Years ago when I was studying liver cancer, we found that that Native American populations also had high rates of liver cancer. So have you looked in the populations you're studying to look at ancestry at all to see whether that explains some of the difference in rates?
Not yet, but we hope maybe with this survivorship study that we can begin to look at some of those issues. Right now, this is all the data that's based on our registries that we've been utilizing. And we didn't have that when we did the small pilot study on liver cancer. But that's a great point. And we do know that we have a fairly large Mexican-American population with Native American heritage.
Thank you.
Thank you. Yes, Luis.
Amelie, if I may follow up with Elena, because this meeting, one of the aims is to identify gaps and to, maybe give some suggestions to our friends from the NCI. I mean, what she's mentioned is a big gap we have, particularly in regions with high Latino population, California, Texas, Florida. So, an approach that we could take is to support through supplements the cancer centers, so we could do this in our population. So, if we're at some point, we'll be talking about ideas, about gaps and about how to fill those gaps. There are cancer centers with a majority Latino populations. Most of them are in 3 or 4 states. There is where we could actually work with our catchment areas and our community partners to do, to try to have a survey that is as close as population-based as possible.
Great, thank you very much. There was one more question regarding childhood cancers. Is there any data on infant formula versus breastfeeding regarding childhood cancer? That's our last question, and then I'm going to take it back to Tram to close this out.
I guess I'll take that one.
Yes.
Yeah, there have been epidemiological studies in ALL, acute lymphoblastic leukemia, that have shown breastfeeding to be protective against ALL. I can't recall off the top of my head if there was any differences, say between Hispanic and non-Hispanic whites, but definitely in general, there's a protective, there's been shown to be a protective effect, and it's thought that's to do with, passing along of some maternal antibodies and regulation, natural regulation of the immune system and immune development that goes along with, some of the theories for the etiology of childhood ALL. But yeah, I'm not sure about other childhood cancers.
Okay, great. Thank you very much. Wonderful panel. Thank you very much for your presentations. Very interesting and informative. Tram?